Hello dear student, I am Dr. Farheen Shahbaz. Basic amino acid. It is from the protein metabolism. Today I am starting from the arginine and referring the book Harper and Mackey Tudor Mackey. So you also follow this book. Now arginine, histidine, lysine, these are basic amino acid, and one by one we will discuss all these amino acid. Today we are only focusing on the arginine. Metabolism of arginine. Arginine is a basic amino acid, semi-essential amino acid, and glucogenic amino acid. Arginine. Arginine can obtain or can generate the glucose, protein. When it is degraded, it can form the creatine, nitric oxide, and the urea. Arginine can be, again, this is the same, the synthesized protein, creatine, nitric oxide, urea, and glucose, whereas now arginine can be entered another fate, that is the ornithine production. Via the ornithine, glutamic semialdehyde will be formed, and from the semialdehyde, glutamic semialdehyde glutamate will be formed. From the ornithine, putrescine and the spermidine can form. These are polyamines, and I will tell you one by one in my coming slide. Now the glutamic semialdehyde forming the proline, whereas proline is a protein, so from the arginine we can say that this protein will be formed, the end product is spermidine, whereas from the glutamate glucose will be formed. Glucose having a carbon skeleton, so it is enter into the TCA first of all, and then again it is going to for the energy production, or it is also help into the gluconeogenesis pathway. Now synthesis and degradation of arginine. In this, from the glutamate, arginine can be synthesized. Now how, look at this, glutamate NADH plus H plus can be formed glutamic semialdehyde and NADH release. From the glutamic semialdehyde, ornithine can form, and also keto acids form in the presence of PLP, and also known as vitamin B6. You all know about that. From the ornithine, arginine can be synthesized or generated. So arginine is resynthesized the ornithine, and if it can be entered the urea in the presence of water and arginase enzyme, so the ammonium ion is entered into the urea. Nitric oxide. Nitric oxide can be produced from the arginine. Synthesis of nitric oxide. Arginine. Arginine can be synthesized citrulline, and in the presence of oxygen, NADPH plus H plus, and the nitric oxide synthase enzyme that is required for this reaction, in which the not only the nitric oxide will produce, citrulline also will form, and the NADP plus will be releases. So nitric oxide synthase require the coenzymes, that is very important coenzyme like FMN, heme, NADPH, and the tetrahydrobiopterin. These four coenzymes required to activate the nitric oxide synthase to release number one nitric oxide synthase to release the nitric oxide, as well as citrulline and NADP will be produced. This slide basically related with the glycine metabolism, but because of course, arginine. So if you look at this, so arginine can be synthesized, the ornithine, and you know that this is a reversible reaction and can be synthesized from the ornithine, arginine can be formed. So from the arginine, of course, other product will be help in the formation of arginine, whereas in an ornithine, the ammonium ion is enter into the urea cycle. From the gonidoacetoacetate, from the glycine, and there by the transaminase reaction, gonidoacetate will be formed, and then it is converted into creatine, and in the presence of kinases, creatine phosphate will be formed, and into the muscles, creatinine can form, that is excreted in urine. So we will be discuss this creatine in the synthesis of or biosynthesis or catabolism of glycine. So when we will learn the glycine, then we will be more focusing on this. Here again the nitric oxide. So it is a toxic pollutant of air, but in human it has a potential biological function. Nitroglycerin is a therapeutic drug for the angina pectoris. Endothelium drive releasing factor EDRF, which causes the nitric oxide. So nitric oxide has a variety of physiological response, potent vasodilator, inhibit the platelet aggregation in immune response in the macrophages. There are different type of macrophages, but here is a microbicidal because it is INOS, and whereas an act as a neurotransmitter in brain and ANS, whereas also act into GI and urogenital tract, stimulate release of gonadotrophin releasing hormone and other important function, as well as are very important again, that is a smooth muscle relaxation occurs. Here is metabolism of polyamines. As we earlier told you that arginine can form the polyamines and is an organic compound having more than 2 amino groups. That's important. Now polyamines possess multiple amino groups. Putrescine, spermine and spermidine are biological important polyamines. Spermine and spermidine are detected in human semen, and that is a very important, that it is present in DNA and RNA, or we can say it's association with that. Actually it's help in the synthesis of the majority of the cell synthesis. So cell differentiation, or we can say proliferation, help in that. Polyamines are basic in nature and possess multiple positive charges. They are associated with nucleic acid, DNA and RNA. They are involved in the synthesis of DNA, RNA and protein. Its concentration is increased in cancers, so it's used as a biomarker in the cancers, or we can say used in a cancer detection. Here this is arginine, and arginine having can convert into ornithine or to the agmatine, but this is into the bacteria plant, but we are only focusing here into the arginine is converted into ornithine by the arginase enzyme, and via the ornithine decarboxylase enzyme it is converted in putrescine. Look at this, there is a 1 amine and here is a 2 amine. From the putrescine, decarboxy S-adenosine methionine, which can be converted via the S-adenosine methionine, and with the help of this and synthase enzyme SPD, spermidine synthase, this is putrescine is converted into spermidine, and via the spermine synthase, and with the collaboration of course with the associated decarboxy S-adenosine methionine, spermine will be formed. In the spermine there is a 4 amino groups or nitrogen groups, and here is an again resynthesis of spermine, spermidine, spermine N1 acetyl transferase. This is also known as SSAT. This SSATs convert the spermine into N-acetyl spermine via the polyamine oxidase SST. It's converted into spermidine, and spermidine via SSAT it is converted N-acetyl spermidine, and via again PAO it is converted into putrescine. So regeneration of putrescine again and again, and these are into the cells proliferation, help in cell fusion present in the cells. But here the difluoromethyl ornithine DFMO inhibit, it's an anti-cancerous, it's a chemotherapeutic drug, anti-cancerous or chemotherapeutic drug, which is inhibit the ODC, because that is helpful for production of all this. So in the cancerous we cannot require more and more proliferation of the cell, so the difluoromethyl ornithine is inhibit the ODC, because it's inhibit the production of these biochemical compounds which is not required, and inhibit the cell proliferation. That's very important biomarkers. Now I will be upload a new video for my students, that histidine metabolism, in next video. And thank you. Ask question and comment.